Welcome to this lecture on reproductive biology. Today, we are going to explore the fascinating topic of lactation and its neuroendocrine regulation. For a newborn mammal, breast milk is the sole source of nutrition and a critical lifeline for survival. But how does the mother's body know exactly when to produce milk and how much to make? The answer lies in a beautifully orchestrated communication system between the brain and the mammary glands. Let us dive in and uncover how this remarkable biological process works. To begin our journey, let us imagine a highly efficient, custom-order food delivery factory. This factory only operates when a customer actively places an order. In the human body, this factory is the mammary gland, and the customer is the newborn baby. When the baby is hungry and begins to nurse, they are essentially pressing a place order button. This sends an urgent electrical message straight to the mother's brain. The brain then responds by releasing special chemical messengers that tell the factory to manufacture the food and pump it out. If the baby stops ordering, the factory naturally slows down and closes its doors to save energy. This demand-driven system ensures the perfect amount of nourishment. Now that we understand the basic factory analogy, let us look at the specific physiological stages of this process. Lactation is divided into several distinct phases. First, we have mammogenesis, which is the anatomical development of the mammary glands themselves. Next is lactogenesis, marking the actual initiation of milk secretion. Once milk production has started, galactopoiesis takes over, which is the ongoing maintenance of this established milk production. Finally, we have the milk ejection reflex, which is the active release of the milk. Each of these stages is critical, ensuring that the mammary glands are properly built, activated, maintained, and able to deliver nutrition to the offspring following childbirth when delivered. Moving from the stages of lactation, we must ask how the body controls all of this. The entire system is tightly governed by a neuroendocrine reflex arc. This means it relies on a partnership between nerve impulses and circulating hormones. The two primary hormones involved are prolactin and oxytocin. Prolactin is secreted by the anterior pituitary gland, and its job is to stimulate milk synthesis in the alveolar cells. Meanwhile, oxytocin is released from the posterior pituitary gland. Oxytocin causes the contraction of myoepithelial cells surrounding the alveoli. Together, these two hormones ensure that milk is not only created in the glands, but also actively pushed out into the milk ducts, where the infant can access it. Let us visualize this neuroendocrine reflex arc with the diagram on your screen. Here, you can see the physical act of the infant suckling at the nipple. This action triggers sensory nerve impulses that travel up the spinal cord directly to the mother's hypothalamus in the brain. In response, the anterior pituitary gland, shown in the diagram, releases prolactin into the bloodstream to command the alveoli to synthesize new milk. Simultaneously, the posterior pituitary gland releases oxytocin, which travels to the breast and forces the milk out of the alveoli. This visual perfectly captures the dual hormone response that translates a physical stimulus into a biological output. Having seen how the reflex arc functions, it is important to understand why this specific mechanism is so vital. Understanding lactation is absolutely critical in reproductive biology because it represents the primary evolutionary survival mechanism for mammalian newborns. Breast milk provides easily digestible nutrition and vital passive immunity through maternal antibodies, especially in the early milk known as colostrum.
Without this, infant mortality rates would be catastrophically high due to malnutrition and infection. Furthermore, the hormones involved play a crucial role in the mother's postpartum recovery. For instance, the oxytocin released during nursing causes the uterus to contract, helping it return to its pre-pregnancy size and reducing the risk of dangerous postpartum hemorrhage.
This biological necessity leads us to a mathematical question. Why must a newborn feed so frequently? Every time the baby nurses, the mother's prolactin levels spike. But once feeding stops, this hormone concentration drops exponentially over time. We can model this biological decay using a standard exponential decay formula. In this equation, P of T represents the concentration of prolactin in the blood at any given time after feeding ends. P0 stands for the initial peak concentration immediately after nursing. The constant E is the base of the natural logarithm, modeling continuous decay. The variable K is the decay constant, dictating how quickly the body clears the hormone, and T is the time elapsed. If time increases too much without feeding, prolactin drops to baseline, stopping milk production. Now that we have our exponential decay model, let us apply it to a real-world scenario. Suppose a mother has a peak prolactin level of 200 nanograms per milliliter, and her decay constant is 0.015 per minute. We want to find her hormone concentration exactly 60 minutes after feeding. We start by plugging our known values into the formula. We place the decay constant and the time directly into the exponent. Multiplying 0.015 by 60 gives us an exponent of negative 0.9. Evaluating the exponential term tells us that roughly 40.6% of the hormone remains. Multiplying this by our initial peak of 200 gives a final concentration of 81.2 nanograms per milliliter. This shows her levels are still elevated enough to signal milk production. So we have proven that frequent feeding keeps hormone levels high. This brings us to the broader concept of supply and demand regulation. Because hormone levels naturally decay between feedings, regular nursing intervals are biologically necessary to keep the factory running. The total daily milk production of a lactating mother is directly proportional to the number of feeding sessions and the average volume of milk transferred per session. This is a perfect linear relationship. If the infant demands more food by nursing more often, the maternal regulatory system automatically upregulates to meet that demand. This ensures that the physical output of the mammary glands perfectly matches the infant's developmental needs over a 24-hour cycle. To quantify this supply and demand relationship, we use a simple linear equation. This foundational formula helps us understand maternal output. The equation states that the total volume, represented by V total, is the product of the number of feeds and the volume per feed. The variable N stands for the number of feeding sessions over a 24-hour period. The variable V feed represents the average volume of milk transferred during a single feeding session. By multiplying these two variables, we can easily scale up a single feeding event to represent an entire day of milk production. If the number of feeds increases, the total volume increases proportionally, perfectly representing the mechanical regulation of lactation. Before we move on to energy, let us see this volume formula in action with a concrete example. Imagine a mother whose mammary glands produce an average of 90 milliliters of milk per feeding session. If her infant nurses 8 times over a 24-hour period, what is the total daily milk volume produced? We simply substitute our given values into the equation. We multiply the 8 feeding sessions by the 90 milliliters produced per session.
This straightforward multiplication gives us a final result of 720 milliliters. This volume represents the total physical output of the mammary glands over a full day, directly driven by the infant's feeding frequency.
Now that we have calculated the total physical volume, we must convert this quantity into the total energy provided to the infant. We do this using an energy conversion equation. In this formula, E total represents the total daily caloric intake for the infant. This is the pure nutritional energy they receive. V total is the daily milk volume we just learned how to calculate. Finally, C vol is a known biological constant, representing the caloric density, specifically the number of kilocalories contained in every single milliliter of human milk. By multiplying the total volume by this density constant, we translate a liquid volume into a precise measurement of life-sustaining energy. Let us apply this energy conversion formula to the volume we found in our previous example. We know the total daily milk volume is 720 milliliters. We are given that human milk contains 0.65 kilocalories per milliliter. We want to find the total daily energy intake for the infant. We substitute 720 for the volume and 0.65 for the caloric density into our equation. Multiplying these two numbers together yields a final answer of 468 kilocalories. This result physically demonstrates how the neuroendocrine regulation of lactation successfully translates the infant's behavioral demand into a highly specific nutritional output. We have covered a lot of ground today, so let us review the key takeaways. First, lactation is a demand-driven neuroendocrine reflex arc, relying on prolactin for milk synthesis and oxytocin for milk ejection. Second, hormone levels decay exponentially between feedings, making frequent nursing essential to maintain supply. Finally, the total daily milk volume and caloric output scale linearly with the infant's feeding frequency. You now understand both the biological mechanisms and the mathematical models behind lactation. This beautifully orchestrated feedback loop ensures that a newborn receives precise, on-demand nutrition, perfectly aligned with their developmental needs.